The primary structure of a protein is the particular sequence of amino acids in a protein chain, and they're held together by peptide bonds. So here is the peptide bond with carbon and nitrogen right here. So that's the amide bond that is being formed when the amino acids come together. So the hydroxyl group, or the oxygen that is connected to the carbon in the carbonyl group, is removed, and then the nitrogen from the other amino acid comes in and forms that amide bond. And the peptide backbone is the amino acid group, the alpha carbon, and the carbonyl group. So this would be referred to as the peptide backbone right there. And then the R groups are substituents along the peptide backbone. So you can see the R groups are right here. And on the other side are hydrogens that attach to the alpha carbon. The peptide chain isn't linear like we just saw in that previous picture, but it has a zigzag arrangement because of the tetrahedral geometry of the alpha carbon. So you can see right here, okay, here's our alpha carbon, and it has a tetrahedral arrangement. So that's that, and you can see around nitrogen and around the carbon and the carbonyl group, those have trigonal planar arrangements, geometries. So that's why it's in that zigzag formation and not just straight across. So the structure of the protein is determined by the amino acids being in the correct order. This primary structure is so important that changing just one amino acid can drastically alter its biological properties. So to see, look at this example here. We have oxytocin here. Nope. Sorry. Okay. And we have vasopressin down here. And you can see the only place that it's different are these two amino acids. And these two have completely different functions. Oxytocin is what causes contractions during labor, while vasopressin, it participates in blood pressure control. So just changing those two amino acids on this peptide chain completely alters the function of that protein. So the arrangement of the amino acids is very important. The secondary structure of a protein describes the type of structure that forms when amino acids form hydrogen bonds between the backbone atoms. So the hydrogen bond is between the oxygen on the carbonyl group, and then the hydrogen on the amide atom, or the amide group. So I'll just show you here on this slide. So here's the oxygen on the carbonyl group, forming that hydrogen bond with the nitrogens, hydrogen there. So these little dots, that's the hydrogen bonding taking place. Okay, so there's two types of secondary structures, and it's the alpha helix and the beta sheet. And we're going to look at exactly what those are. Okay, so first the alpha helix forms a coiled shape by hydrogen bonding with, again, the nitrogen. It's the hydrogen that's attached to the nitrogen group in one turn, and then the oxygen from the carbonyl group in the next turn. So you can see right here. So we have this nitrogen. It's kind of hard to see. That nitrogen is hydrogen bonding, or the hydrogen from that nitrogen is bonding with the oxygen in that carbonyl group. So same thing down here. Here's the oxygen in the carbonyl group, and then there's the hydrogen that's attached to the nitrogen. So they're in different turns. So that's what holds it together in that arrangement, in that coiled shape. Okay, then we have the beta sheet. The polypeptide chains are held together side by side by hydrogen bonding that forms between the oxygen atom and the carbonyl group, and in one section of the polypeptide, and then the hydrogen atom in the NH group, so like we've been seeing, but in a nearby section of the polypeptide. So it can be the same polypeptide, or it can be two different polypeptides. So again, here's the nitrogen with the hydrogen, forming that hydrogen bonding with that oxygen. Okay, so this can be the same polypeptide. If it was, it would just kind of, do a little turn there and wrap around and do another turn. So that's what it would look like if it was the same polypeptide. And now we can make the beta sheets with, two different peptides as well. So anyway, just wanted you to see that that it can kind of wrap around and then form those hydrogen bonds. Okay, proteins are classified in several ways, and one of them is to identify them as fibrous or globular proteins. So fibrous proteins consist of long, thin fiber-like shapes. They're typically involved with structure of cells and tissues. And there's two types of fibrous proteins. They're alpha keratin or beta keratin. So fibrous proteins can have the alpha helix or the beta sheet structure or arrangement. And then for globular proteins, they have a compact spherical shape because sections of the polypeptide chain will fold over on top of each other due to the interactions of the R groups. So globular proteins have functions in the cells like synthesis and transport, metabolism. And then I have a couple examples here for you from your book. So the spider web is an example of a fibrous protein, and then the cheese and the milk is an example of a globular protein. There's some more examples here. Specifically the name of the proteins. We have keratins, collagens, elastins, myosins, and fibrin. All of those are fibrous proteins. And again, you can see, we just mentioned that fibrous proteins are typically involved with structure of cells and tissues. And you can see that in the function that's listed here for each of those. Okay, and then we have globular proteins. We have insulin, ribonuclease, immunoglobulins, hemoglobin, albumins. So just some examples there of fibrous or globular proteins. Okay, the tertiary protein structure involves attractions and repulsions between the R groups. So, in secondary structure, we were looking at just hydrogen bonding between really the peptide backbone. But in tertiary structure, it's all about the side chain interactions. So as these interactions occur, the protein is folded into a specific 3D shape. And the way the protein is folded is determined by the primary structure. So those amino acids in the primary structure, depending on what the R groups are, will determine what 3D shape the tertiary structure is going to form. So here's a look at the tertiary protein structure, ribonuclease, and you can see it has a mixture here of alpha helix, beta sheet, connecting loops, disulfide bonds. So all of those are formed in stabilizing this tertiary structure. So we're going to look specifically at what interactions are taking place. So these are the ones we're going to look at. We have hydrogen bonds, we have salt bridges, hydrophilic interactions, hydrophobic interactions, and disulfide bonds. So again, the structure, the tertiary structure, is stabilized by all of these interactions that are taking place between the R groups. So this is kind of one of them we're going to refer back to. This picture here shows all of the interactions that we're going to talk about. Okay, so first up we have hydrophobic interactions. So the hydrophobic R groups will cluster together to create a water-free pocket. So remember when we talked about nonpolar amino acids, we talked about how those are hydrophobic. So they don't want to interact with water because they're nonpolar, so they cannot form bonds with water. So what these nonpolar R groups will do is they'll cluster together, and so here's two amino acids that have nonpolar side chains. Okay, phenylalanine and leucine. Okay, but this picture back here really gives a better look at it. So here's that hydrophobic pocket. Okay, they kind of cluster together right there. Here's some hydrophobic interactions. So you would have, like London dispersion interactions taking place between the nonpolar R groups. Okay, and then we have hydrophilic interactions. So the R groups are able to interact with water, and they'll be on the outer surface of the protein. So here we have aspartic acid, and you can see it's able to hydrogen bond with, this is the R group right here, and it's able to hydrogen bond with the water. So those will be on the outside of the protein. I don't believe this picture has any of that on there. But they would be out here, those R groups, so they can interact with the water. Okay, then we have hydrogen bonding. So hydrogen bonding occurs between the hydrogen of a polar R group and the oxygen or nitrogen of another amino acid. So here we have threonine, and it has an OH group. So that hydrogen is hydrogen bonding to asparagines, R group, the oxygen in it, to form that hydrogen bond. Okay, so let's go back here. So we have several examples of hydrogen bonding taking place in this picture. So we have hydrogen bonding here, and then we have the hydrogen bonding that takes place, between the sheets, the beta sheets, and over here with the alpha helix. But then we just have, some of these random hydrogen bonds between the R groups as well. So I guess that's something important to point out. These three here in the middle that I've circled, these are hydrogen bonding between the R groups. But we saw earlier the beta sheet, so here's this sheet, and here's the alpha helix. That's hydrogen bonding between the peptide backbone, not the R groups. So that's a little bit different. Okay, and then salt bridges are ionic bonds that are formed between acid and basic R groups in their ionized form. So the positive and the negative charge will form the ionic bond. So here we have aspartic acid and lysine. So in its ionized form, aspartic acid has this negative oxygen right here, and then the lysine has a positive nitrogen or amino group. So that it's able to form that salt bridge because it has that positive and negative charge. So we're forming an ionic bond between these two atoms. I'm gonna go back here. So here's an example. Let me get a different color. Here's an example, excuse me, of a salt bridge. So we have this positive amino group and we have a negative oxygen. Now they're attracted to each other. Okay. Oh. Last of all, we have disulfide bonds. And disulfide bonds are just covalent bonds. So remember, covalent bonding has to do with sharing electrons, whereas ionic bonding was all about transferring electrons. So they're sharing electrons in these disulfide bonds. And they form between the thiol groups of cysteine in the peptide chain. So here we have 2 cysteine amino acids. And then it's able to, these hydrogens are removed, and it's able to form that sulfide or sulfur-sulfur bond. So let's go back to our picture here. Okay, so we have one right here. Running out of colors. We'll do blue. Okay, so this is the disulfide bond down here. You can see that sulfur right there. Okay, so all of these interactions are taking place, and all of them are specific based on what amino acids are within the peptide chain. It determines what interactions are going to take place, what the tertiary structure is going to look like. So conjugated protein is a protein that is aided excuse me, in its function by the presence of a non-amino acid unit. So an example is myoglobin. So myoglobin has a heme group that's embedded within the polypeptide, and you can see that here in the picture. Right here is the heme group. Okay, and the oxygen, or an oxygen, attaches to that heme group so that it can transport the oxygen. So the myoglobin can transport the oxygen. And there's more examples of different types of conjugated proteins and what is embedded in those proteins. So for example, glycoproteins have carbohydrates embedded in them. Lipoproteins have lipids, and nucleoproteins have RNA. And there's a couple more in table 5 in your book, but just examples of conjugated proteins that are aided again in their function because they have a non-amino acid unit within the protein.